Professor of Molecular Ophthalmology at University College London and the Francis Crick Institute in London. And I'm also a consultant ophthalmologist specializing in genetic eye disease, and specifically inherited retinal diseases, at Moorfields Eye Hospital in London and Great Ormond Street Hospital for children. And I see adults and children with any form of inherited eye disease, but a particularly strong research interest that I have is in Usher syndrome. And so I do see a number of families with this. In terms of my research, it spans from seeing patients in the clinic, trying to understand what gene causes their condition. When we don't know what their gene is, looking a little bit more deeper to find new genes and new mutations, and then looking in disease models to see or prove if those mutations are actually causative. And to do that in my lab, we have various different models that we use. So I use zebrafish, and I have generated a zebrafish with an USH2A mutation so that we can use it to study the disease, but also to test therapeutics. We use patient skin cells, and we can convert those skin cells into stem cells and then grow retinas to resemble early eye development in a dish. And it represents the kind of the disease that is going on in the retina in patients. So again, that helps us have insights into the role of that gene in health and disease, giving insights into disease mechanisms, and then using that model again to test for therapeutics. We also use the mouse model. And then moving on to the kind of clinical research that I do, you know, being fortunate to work at Moorfields, we have one of the largest patient resources in the world. I looked in our database, we have 28 families with a molecularly confirmed ADGRV1 mutation. So that's the cause of USH2C. So that's quite exciting to add to maybe the group of families that you've already got connected to yourselves. But what we like to do from a clinical point of view is undertake natural history studies to study these conditions over long periods of time, retrospectively, to get an idea of how patients progress, what their prognosis is like for the future. And then we undertake prospective studies, so where we bring patients in every, for example, 6 months for maybe a 1 to 2 year period. And we undertake really detailed investigations so that we can identify outcome measures for clinical trials. And then obviously the last stage of my research is undertaking clinical trials. And I'm the principal investigator for the USH2A ProQR trial at Moorfields. And that's an absolute privilege. So a lot of what I'm going to talk to you about is an overview about Ushers. Forgive me if it's repetitive, but I just wanted to kind of set the scene, talk about USH2C, the similarities that it has with the other type 2 Usher conditions, and some of the research. And then I'm very happy to answer any questions. And Michaela, you may want to just ask those questions that were raised by the group through your emails towards the end. So I'm just going to share my slides now. Oh, somebody has to let me share my slides. If someone can.
That's probably me. 1 second. Does that allow you? Can you do it now?
Yeah, perfect.
Okay.
Okay, can you see those?
Yes.
Okay, let me just make that big. Okay. So the title of my talk is USH2C, a clinical and research update. I've given you my introduction as to who I am. And I've already gone over the overview of this talk, but just to recap, to talk about Usher syndrome, USH2C in particular, and to discuss the current research for USH2C and Usher syndrome overall. And then I want to mention a really good web resource called Gene Vision. So Usher syndrome, as you're all aware, is a rare genetic eye disease. Well, it's a syndromic eye disease. It affects between 4 to 17 individuals in every 100,000 individuals. And it is a cause of hearing and vision loss, with a variable presentation of balance problems depending on the type of Usher syndrome that you have. And there are 3 types, type 1, 2, and 3. So just as a summary, patients with type 1 Usher syndrome have the most severe form. They're born with severe to profound sensory neural hearing loss from birth. Their balance is affected, and they start to develop a retinitis pigmentosa, so a retinal degeneration, from less than 10 years of age. And there are several genes that are known to cause it. Type 2 Ushers is the most common form of all the 3 types of Usher syndrome. Again, children are born with moderate to severe sensory neural hearing loss. Their balance is not affected, and they tend to develop the symptoms of retinitis pigmentosa slightly later on in childhood, in their early teens and into their 20s. And there are 3 main genes that cause this: the USH2A gene, that accounts for 90% of type 2 cases, the ADGRV1 gene, which accounts for about 9%, and then WHRN whirlin, which accounts for about 1% of the type 2 cases. And then you've got type 3 Usher syndrome. And this is quite variable. It's gradually progressive in terms of its hearing loss, sometimes from puberty. There's variable presence of balance problems. They do tend to get the retinitis pigmentosa, and there are 2 genes that are known to contribute to that. So usually for type 1 and type 2 Usher syndrome, it's usually picked up on the newborn hearing screen. Usually patients should be referred to ophthalmology at that point. It doesn't always happen, but we, at least in the UK, we do know that there is a protocol in place that patients that are failing that newborn hearing screen are referred to ophthalmology for a review. And in fact, now a lot of children who have sensory neural hearing loss are actually sent for genetic testing at that time for a deafness gene panel, and that actually includes the genes that are known to cause Usher syndrome. So sometimes some children are picked up very early on before they even start to develop any of the visual problems. So when they're referred to ophthalmology, we obviously take a history, and the first signs of any retinal degeneration is night blindness. So patients find it difficult to see in the dark. And that's because this condition initially affects the light sensitive cells, the photoreceptors in the retina that particularly are sensitive to light and dark and to the field of vision. And those are your rods. And then you have your cones, which are usually used for color vision, central vision, near fine detail vision, and they tend to be affected later on in the natural history of the disease. So the initial presenting complaint is difficulty seeing in the dark and possibly some visual field loss. And then we will look at the back of the eyes. And as I said, if it's early and the child's still young, then the retina might look entirely normal, but there may be some early signs of the retinitis pigmentosa. And we do several tests. So we would do visual fields tests, take images of the back of the eye, do a scan to take a cross section of the retina to see what that looks like, do a big flash of blue light into the back of the eye to look at how the back of the eye is fluorescing. And then we can do electrical measurements of the retina. And sometimes that's when we detect that there's a problem with the retina before we can physically see something. And then obviously we want to undertake genetic testing. So this is an image of a healthy retina on the left. And this is an image of a patient with Usher syndrome showing signs of retinitis pigmentosa. And so you can see in the periphery, there are these sort of clumps of black pigment, and these are called bone spicules. So that's something that we might pick up on. For example, an optician might see it in a routine eye check. Well, we would see that in our ophthalmology clinics. Later on, the actual blood vessels that are supplying the retina start to become really thin and thready. And then the optic nerve, which should be a sort of nice pinky yellowy color, actually starts to become quite pale, and we describe it as looking waxy. And this is a patient with quite advanced retinitis pigmentosa. And you can see all the kind of the black pigment in the periphery of the retina where the rods have degenerated. When we do that big bright flash of light in the back of the eye and we measure the fluorescence, these are the kind of images that we see in our patients with retinitis pigmentosa. You can see, just to orientate those of you who can see these pictures, we've got a nerve that sends signals to the brain, we've got the blood vessels, and we've got a big retina. In this peripheral part, you have the rod, light sensitive cells, and in the center, this is your area of central vision, your macula, which helps you to see color and fine detail, that has a lot of cones. And you get this characteristic, really like a white ring around the macula. It's called a hyper autofluorescent ring, and that's very characteristic of patients with retinitis pigmentosa. And we can actually draw around that really bright white ring, and we can measure the circumference of that ring over time to see if it's getting smaller and smaller, and that's a sign of disease progression. I mentioned we can take a cross section through the back of the eye using infrared wavelength of light, and it allows us to see the layers of the retina, and it particularly allows us to see the boundary of those light sensitive cells with the mother kind of supporting cells, which are called the retinal pigment epithelium. And again, we measure that over time to see if it's getting smaller and smaller, because that's again gives us an idea if a patient is stable or how much they're progressing. We undertake visual fields, and this is again just an example of a normal visual field. There's a black area which is where your optic nerve leaves the back of the eye and travels to the brain, and we call that our blind spot. So when you're cycling or you're in a car and you don't see a cyclist and you say, oh, it was in my blind spot, that's where your nerve is leaving the back of the eye, and that's normal. But in obviously any more black than in that small area is a sign that you can't see. And so with patients with retinitis pigmentosa, you get a constriction of the visual fields, and only the area in the center is where you can see well, and the rest is usually black because of the degeneration. We undertake electrophysiology. As I said, you can put electrodes around the head and around the eyes, and you can flash, patterns of black and white squares, bright lights, and you can measure the responses of the retina and how signals are being passed to the brain. And this is really important in younger children when you're not sure if the hearing loss is just isolated or in fact if it is coupled with a retinal degeneration.
So Dr. Moosajee, if you don't mind popping in with a question. So most of our members do have younger children who were picked up via the genetic screening that was done. They had hearing loss at birth and then followed up with genetic. For something like the ERG or any of these other tests that you just mentioned, what would be the ideal age for them to undergo? We had been told, they really can't sit for any of them till they're over 5 years old. But, based on what you've seen and what you do in the clinic, is that what your team recommends, or just so we can provide some guidance to our members?
Yeah, so I mean, to be honest, now we have genetic testing. That's your gold standard. That's your molecular diagnosis. We used to use electrophysiology a lot more before we had genetics because we didn't know what was the cause. We didn't know if it, as I said, it was an isolated hearing loss or not. I think if you've got a mutation confirmed in the gene that causes the USH2C, then you don't 100% need to have an retinogram. However, because with USH2C, you start to get the night blindness later, you don't actually need to have it when you're a young baby. So, I would go with the guidance of around 5. That's absolutely fine. I mean, even if you pick something up, it might be subclinical. And sometimes I know patients who've had it very early on, and it's either equivocal, the child was too young, as you said, they weren't kind of sitting still, they couldn't get a good reading. Or you pick something up and there's nothing you can do anyway. So,
Right.
I don't think it's 100% essential. And also, if you see, I've put that actually you only really need to do this once. This should not be something that you need to have repeatedly. So it's better to wait till the child is a little bit older and more compliant.
So not a 2-year-old.
No, so absolutely, it's absolutely fine to wait until then, if you've got your molecular diagnosis.
Okay, thank you.
I actually, personally, and just this is from an international point of view, again, we're very privileged in the UK because we have a national health service that essentially funds everything. But there are patients from all over the world that don't have access to funds or expertise. And sometimes, they really spend their time gathering all the funds they can so that they can go and have these investigations. And I'm often asked, which is the investigation you would do? Number one, genetic testing, without a doubt. So, that is the biggest piece of advice. Once you've got your molecular diagnosis, that's like your passport. You keep that safe. That will be what you use to get into trials and get treatments in the future. And then I actually just monitor patients with the fundus autofluorescence and the OCT. There are unfortunately, some places you'll go, they'll overdo the amount of tests that individuals need, and also far too frequently. So you actually only need an OCT and an autofluorescence annually, in my opinion. You don't really need it more than that, because you don't tend to see change in less time than 12 months. So I hope that's helpful, just as a guide.
Yes, thank you.
So all the forms of Usher syndrome are inherited in an autosomal recessive manner. So this means that parents are usually carriers of the condition, which means, so when we have a child, we all basically have our own complement of genes. Dad will give half of his genes and mother will give half of her genes. So every child will have 2 copies of every gene. Now, what happens with patients who are born with Usher syndrome is that their father will have a good copy of a gene and a bad copy of a gene. But because he has a good copy, it's strong enough to overcome that bad copy, and he doesn't have any signs of the condition. And he's referred to as a carrier. That same scenario exists for the mother. So mum will have a good copy of a gene and a bad copy. And again, she will be fine because that good copy is strong enough to prevent the disease from showing. But then they have a 25% chance of passing the 2 bad copies to their child. And that's when you start to see Usher syndrome in children. Now, obviously, that can vary slightly. For example, if family members if a parent is already affected, or if there are ancestors who are relatives with one another. But generally, the inheritance risk is a 1 in 4 chance of having a child with Usher syndrome. And then for a patient with Ushers themselves, if they go on to have children, but they marry an individual that is unrelated to them and has no history of Ushers, then the most likely thing will be that their child will be a carrier of the condition, which means that the individual has no option but to pass a bad copy to their child, but the partner would hopefully pass a good copy of the gene, and therefore their child would be a carrier but unaffected. But I would just urge people to have those conversations with their genetic counselors. And if they want to have more children and they know their genetic diagnosis, they can go forwards to have things like pre-implantation genetic diagnosis and lots of different options for family planning. So the importance of genetic testing, because patients, families want to know the cause of their condition. We as clinicians want to know the cause of patients' conditions so that we can give them the correct informed genetic counseling, we can get the right multidisciplinary team involved, we can then give them family planning options, we can study their disease, develop new treatments, and we can give those families access to the latest research and clinical trials. So USH2C, as I said, is caused by mutations in the ADGRV1 gene. And this gene was previously called VLGR1 and GPR98. But ADGRV1 is now the settled name of the gene, and it stands for adhesion G protein coupled receptor V1. And it's found on chromosome 5. Now, this particular gene and the protein that it produces is found in the ear and the retina. And it's known to form a complex with the two other USH2A gene proteins. So the USH2A gene, usherin, and the whirlin gene, and the protein is whirlin. And so basically these three genes express these proteins that come together in a complex, and they are found in something called the ankle links of the hair cells in the ear. And you can see that in my diagram. And these ankle links actually form prenatally when the baby is developing in the womb. And they develop to basically guide the structure of these hair cells so that they develop properly. And then once the hair cells are in their correct position, these ankle links actually dissolve and they're not there postnatally anymore. So this is why children are born with sensory neural hearing loss, because this event happened in utero. In the retina, it's a slightly different story. That complex is found at something called the periciliary ridge. It's right in the middle of our photoreceptors. And there's a diagram here, and it's where I've got the little red area. It's basically between what we call the inner and outer segment of the photoreceptor. And it's thought to be a docking site for key proteins that need to move from the inner part of the cell to the outer segment of the cell. And one of those key proteins is called rhodopsin, and the opsins, the light sensitive pigments that are excited in our photoreceptors that convert light energy into chemical energy, which is then passed down through our retina and through the nerve to the brain. And so what we found is that these opsins are not transported to the right place, they're mislocalized, and I'll show you that in some of the work. So I mentioned that USH2C accounts for around 9% of all type 2 Ushers. And from various databases, we know that there are 168 recorded mutations in this gene, compared to, for example, in the USH2A gene, where there are over 1,000. And this is an incredibly large gene. In fact, all of the genes for type 2 are big. But USH2A and ADGRV1 are very big genes. And ADGRV1 is actually 19,500 nucleotides or bases or letters long. It's a massive gene. And so, you know, thinking about things like gene therapy it's quite tricky with these big genes. But hopefully I've got a solution for you as I proceed later on in the talk. So recently a study was published actually looking at a comparison between USH2C and USH2A. It was published by Anna Faking and colleagues from Slovenia last year, and they looked at 18 ADGVR1 patients compared to 204 USH2A patients. And then interestingly, they didn't find much difference in the age of onset of the retinal phenotype, so the retinitis pigmentosa. In fact, they found, I mean, if you look at the graph that I'm showing, you could suggest that maybe USH2A patients actually had an age of onset slightly earlier than USH2C, around, I think their mean age was 18 years. Whereas the ADGRV1 patients, the onset was more closer to 30 years. But when we looked at the standard deviation, it wasn't statistically significant. Those error bars are quite big. They didn't find a difference in the rate of progression between the two sets. And also they looked at the development of macular edema, so fluid at the macula, the central area of the eye. And again, they found no difference between the two groups. And they actually concluded that actually they couldn't tell a difference between the retinas of an USH2C patient and an USH2A patient. And I think that's actually a good sign, especially because of all the developments in USH2A. It gives us a great idea into the kind of the window of therapeutics, the kind of outcome metrics we should be using, so we can learn a lot from USH2A and apply that to USH2C going forwards. So in terms of the research, there's lots and lots of research going on. This is an amazing time for people like myself in genomics and genetics, but also for patients, because, you know, you've just said that most patients are getting a genetic diagnosis before they're even getting retinal symptoms. And that means that if these therapies actually come to fruition and become approved therapies, our kids can actually access this before they even start to get retinal degeneration or at a very early stage. So there's lots of things under development in this space: gene-based therapies, gene editing, RNA therapies. And then there are lots of therapies looking at common disease pathways. So drugs such as antioxidants. We know in the back of the retinas of patients with RP, there's a lot of oxidative stress going on. And so we need to neutralize that with antioxidants. How can we target the cell death pathways using neuroprotectant drugs? Gene therapy. So I mentioned that the rods die off first. Rods are known to secrete something called rod-derived cone viability factor. This is a factor that helps cones uptake glucose and convert it to energy and work efficiently. And so when your rods die, your cones can't uptake that factor anymore, and they essentially starve to death. And so if you can replace this rod-derived cone viability factor back, even if you've just got cones left, it might be able to preserve vision for longer. So that trial is underway. The use of stem cells is underway too. The current trials for Ushers that are active, there is a trial for a high-dose antioxidant, NP1001, and that's underway in Australia. That's due to complete in September 2023. It's a phase 1/2 trial, and they're looking for 48 participants over the age of 18. And one piece of advice that I give all my patients with RP is please have lots of fresh fruit and vegetables in your diet. The vegetables that are green, leafy vegetables, kale, broccoli, spinach, salad, peas, anything green. And then in terms of the fruit, the best fruit are those that are red, blue, and black in color, because they are rich in antioxidants. And so things like blackberries, blueberries, raspberries, strawberries, pomegranate, black and red grapes, black and red plums, raisins, all great for you. So please have lots of that until we find out the results of these trials. The next thing is there is a phase 3 study. Oh, I've got a typo there. It's Sirius, not Syrian, that I put there. And this is an RNA therapy that is underway with ProQR, where basically they are injecting a short strand of nucleotides into the eye, and it binds to exon 13 of the USH2A gene, and it prevents on the instructions, not on the gene or the DNA, but on the instructions, which are called RNA, and it prevents that exon 13 from being kind of translated into the protein. And that's because there are a lot of common mutations in that exon. Now, we don't know whether that's going to work effectively or not, because there are patients that have mutations where the whole of exon 13 has been removed and they still have Ushers, but we're going to wait and see. We should know the results in the next 2 to 3 years, but they are currently recruiting patients 12 years and upwards, and we are keeping our fingers crossed that it does work. Previous trials, well, there was a trial for MAIA-7A, a form of type 1 Ushers, and that was using a virus that was not the traditional one that's been approved. It was something called a lentivirus, and that is thought to be quite immunogenic. It causes quite an immune reaction, and so that was actually terminated because of safety concerns, and so that's a real shame. And then there was recently another trial that was looking at injecting ciliary neurotrophic factor, which is a neuroprotectant, a drug that will support cells and prevent them from dying. And that actually closed in 2019, but so far we haven't heard the results yet. So, you know, hopefully we might hear at some point, I hope. So just kind of in the last few slides, I want to just talk about my own research. I mentioned that I undertake a lot of clinical trial, a lot of natural history studies at Moorfields, because we just have a wealth of patients. But I've undertaken natural history studies for USH2A and MAIA-7A, where we've brought patients in at baseline 6 months and a year, and we've done a multitude of tests. And what we found is parameters that we can then use in clinical trials that show a difference over a certain time period, whether that be through OCT or fields or microperimetry or OCT, or using a combination of different imaging techniques. But this is certainly something that we could do for our USH2C families. And I always urge groups where we have these well-characterized phenotypes that we should be doing this now, because if a treatment does come to fruition, we want to be ready. We don't want to be starting these studies later on down the line and delaying everything. From a basic science point of view, again, I mentioned my zebrafish. We've created a zebrafish model with USH2A. We use gene editing to do this. And when we studied it in a lot of detail, what we found was that light-sensitive pigment, rhodopsin, was not being expressed in the right place. And you can see that in this image, that in a normal healthy zebrafish at 12 months, all the red staining is within the outer segments of the photoreceptors. But in the USH2A, it's kind of dotted all over. And that's because it hasn't transported to the correct place. And then we looked with really high ultrastructure resolution, and we found that there were this increase of these bodies within the light sensitive cells, which are called lysosomes, which are used to break up unwanted material. But there was loads of these structures. And so what we found was that actually, instead of this light sensitive pigment being transported to the right place, it was clogging up the cell, and the cell was producing more of these lysosomes to break up all of this kind of buildup of unwanted material. And then we also found that there was an increase in something called autophagy, which means the cells are basically triggering a response to digest themselves. They were self-eating. And so this really gives ideas and targets for developing treatments that may help patients with Usher syndrome. We can from patient skin cells. And we do this to help us understand the disease and use it to test therapies. And so these are just some images of a patient's cells who has USH2A mutations. We've got what we call an induced pluripotent stem cells, and these stem cells can be converted to any type of cell essentially, but we've just grown early eye cups so that, as I said, we can study the disease. So there are limitations with viral gene therapy. First of all, there is a risk of introducing further mutations into one's genetic code. There's a significant risk of triggering an immune response. The gene therapy may stop working. And the big limitation for USH2C is that viral vectors can't accommodate large genes. And so they have a limit of 4,500 bases or letters, whereas, as I mentioned, the USH2C gene is 19,500 bases. So it's just no way going to be able to fit into a conventional virus. So in my lab, what we're doing is working with non-viral gene therapy. And these are essentially human DNA, circular DNA, which can hold unlimited sizes of genes. The largest gene that it has held has been 135,000 bases in size. I mentioned the USH2A gene is 6,000 and ADVRG1 is 19,000. So there would be no issues with getting the USH2C gene into one of these vectors. They're composed of entirely human elements. There are no viral components, so there's no associated immune reaction, and it doesn't integrate within your DNA. And it's got this special ingredient, which is called a scaffold matrix attachment region. And this basically allows this plasmid vector to be stable within our photoreceptor cells and allow it to produce the gene of interest for really long periods. So we've done a lot of work with USH2A, and we've produced a non-viral vector with the USH2A gene. And the way we had to do this is cut the gene into small pieces and individually place it within this vector like a jigsaw. And then we sequenced it to make sure that we had all the letters all in the correct position. There was no spelling mistakes whatsoever. And then what we did is we delivered this vector to our zebrafish model and also to patient skin cells. And the amazing news was that we saw gene expression, so production of the USH2A gene up to 12 months in the zebrafish retina after a single injection. And we found it expressing in our patient skin cells after several different rounds of division and passaging. So now what we're doing is testing these vectors on higher order animal models. So there's a rabbit USH2A model, but also in pig models to look for safety, because that's the closest biomedical model that's similar to the eye. And this is just an interesting piece of results from the work. But when we looked at the retina of our USH2A zebrafish, we actually saw loss of the ADGRV1 protein. So what it's showing here is if you don't have usherin to form a complex with ADGRV1 and whirlin, you don't get the presence of that protein in the Usher retina. But when we delivered the USH2A gene therapy, we saw restoration of ADGRV1. And so, you know, the fact that this has worked for USH2A, you know, I'm pretty hopeful that this would also work for ADGRV1 if we were to push forwards with an application for this gene for this kind of non-viral gene therapy. And the last thing I just want to leave you with is this amazing resource. It's called Gene.Vision. We launched it in December 2020 from the UK. It was created by myself and my colleagues. And it's basically a resource on all genetic eye disorders, but there's great pages on Usher syndrome for both patients and professionals. So the patients' pages are fully accessible, and it's written in lay language. It works with all speech softwares, but there's also a page for professionals and explains all these new research therapeutics in an accessible format for you. So please do look at this and promote it to individuals all around the world who might be looking for a reliable resource to get the latest information from. And this is just what the Usher syndrome page for patients looks like. So how can you get involved? Well, as I said, if you do have the option to enter into natural history studies or prospective or retrospective, to use your clinical data, please allow us to access it. It really will help us prepare for clinical trials. If any of you want to get involved in the research, are willing to give skin samples, please get in touch. Fundraising is a massive thing. Unfortunately, we have all these amazing options that we could apply to USH2C, but we really need research grants to support the work. And I'm very happy, to liaise with you or talk to you if that's something that, you're interested in doing. And just keep in touch. I've put my email address there. It's m.moosajee@ucl.ac.uk, but Michaela can send that out to everyone. And I'm happy to take questions now. Do follow us on Twitter, where, we post all our work and our latest papers. And just a big thank you to my group, who've done amazingly well to push forwards research into Ushers and all our funding bodies. Thank you so much. Thank you.
64 and pretty well lost the last of my useful vision last year. And I'm aware, my perception of retinitis pigmentosa type diseases is that the older you are and the less healthy the back of your eye is, the less receptive you are going to be to any potential cures or trials. Is that a rule of thumb, or is that wrong?
Yeah, so unfortunately, at the moment, in the kind of current playing field of research, if you have lost your light sensitive cells, they can't be regenerated. So it's sort of what's lost is lost. However, I mean, scientists are just amazing. So there's a new form of gene therapy under trial at the moment called optogenetics, which is basically where they are injecting a gene that will produce a light sensitive channel that will integrate into the remaining cells in your retina. And so in certain wavelengths of light, those light sensitive channels will get stimulated and can send signals down your nerve back to the brain. And it's not perfect vision. It's not like how you were sort of, 20 years ago. But if if you're at an advanced stage, and we're talking about light perception, then it may potentially give you navigational vision. You may be able to see outlines and shadows. We have to wait to see. But yeah, and then, with further advances with stem cells and regeneration, that might become an option. There's obviously the bionic eye, where they put a microchip at the back, again, sending signals down the nerve. So they're very crude still, but this work is going on.
Jumping off of that question, I know you just spent some time going over what the current trials are looking like in research. There's a parent of a kid who's 5 years old that was just diagnosed from genetic testing. What can they be expecting in terms of any sort of a general timeline of when these trials may, continue forward and be applicable in terms of treatments for their child?
Yeah, so I mean, if you're talking about, say, now finding a diagnosis or waiting for these outcomes, I would like, gosh, everyone always says this, don't they? 5 years. I'd like to say 5 years, but then you've got, for example, one of the studies I said closed in 2019, but it's been already 3 years and we haven't heard the outcome from it, which probably may suggest that it hasn't been successful. Now, all I would say is that the industries are pumping a lot of money into this field in terms of genetic therapies, and they spend a massive amount of money on clinical trials. So it's only in their interest that if a clinical trial at phase 3 shows that it's worked, that they get that to market approval as soon as possible. And so, you know, I'm confident that if, for example, a 5-year-old came to my clinic and they asked me that question, I would say in the next 5 to 10 years that there's going to be something, whether that be, for example, those high dose antioxidants, or maybe something that's just going to slow. It may not be a gene therapy right now, and especially with USH2C, I'm not aware of anything that is happening with USH2C at the moment, in which case, you know, it may take 10 years before we get to a point of, you know, a clinical trial or results from a clinical trial. So yeah, I would probably give a guide of about 5 to 10 years, but based on that, you're still going to have quite good usable vision.
And that actually reminded me of the other question that I received multiple times, is this whole debate about vitamin A. So I know you said, you know, leafy green vegetables and black, red, and blue fruits. What is your take on vitamin A?
So all the consultants at Moorfields do not give or advise vitamin A supplements. So the reason that vitamin A is such a popular topic, it's because about 30 years ago, somebody identified vitamin A as a key part of the visual cycle. And vitamin A is changed into different forms as it circulates through the light-sensitive cells and the retinal pigment epithelium. And so back then, everyone used to say, yes, have lots of vitamin A. And then what happened was a subset of patients started to lose their vision and go blind. And they were the patients with Stargardt's disease, a mutation in ABCA4. And it's because their mutation meant that they couldn't process vitamin A, and then they got toxic levels of vitamin A in their retina that caused death, cell death, and accelerated their condition. Now, that isn't the case for RP patients. However, our general consensus is that you should not be taking vitamin A supplements, because you don't need excessive vitamin A, because too much is going to be toxic. What you need is a healthy, balanced diet where you can include it in normal levels, but not take too much. And I know there was a trial in Australia that suggested this, but actually it wasn't there wasn't enough evidence base to say it was successful. So I would just say healthy, balanced diet, full of fresh fruit and vegetable. Have fish twice a week. Fresh fish twice a week is really good for you, because they say that tablets with omega oils have been sitting on shelves and potentially oxidizing, whereas fresh fish would be much more beneficial to go straight in, until we have a lot more solid evidence base.